Morning students of class 10, welcome back to the online bio classes. In the previous class, we had already started the first chapter, that is cell and cell division. You remember what we had studied in the previous class? It was definition of cell and the types of cell division. Cell division is the process by which the parent cell produces daughter cells. And there are mainly two types of cell division found in the living organisms. They are mitosis and meiosis. Mitosis cell division is found in the somatic or body cells, also known as somatic division or vegetative division. And the second one, that is meiosis cell division, is found mainly in the reproductive cells, also known as gametes. They help in the formation of gametes. So in today's class, we are going to study about, in the previous class we studied about the cell cycle also, the diagram of cell cycle and all the three stages, that is G1 phase, S phase, G2 phase, M phase. Cell cycle means the sequence of events which occurs during the life of a cell when it undergoes division. Like not only the nuclear changes occurs, but the cell also grow in size, growth of cell occurs, changes occurs in the number of chromosomes, and it also results in the formation of new materials inside the cell which are required for division. Then we talked about G0 phase. G0 stage refers to that part of the cell cycle in which some of the cells, like our brain cells or the heart cells, which fail to divide, they will exit the cell cycle. Means they are not going to take part in the remaining part of the cell cycle. So let's continue with today's class, that is types of cell division. Types of cell division. Broadly two types are there, but here we are going to talk about three types of cell divisions are there. I just draw it like this. Number one, amitosis. The word A is used. It is a simple division. Second one, mitosis. And the third one is meiosis. You don't have to study the stages of meiosis, it's not there in your syllabus. Broadly you have to study this one along with the diagram, this one only definition and some characters. So there are three types of cell division found in living organisms. So let's study with the first one, that is amitosis. It is a simple division. It is a simple cell division in which there is no changes in the number of chromosomes, in which there is no changes in the number of chromosomes, and there is no spindle formation, and there is no spindle formation. Spindle fibers are also not formed here in this type of simple division. What happens? First of all, in this mitosis cell division, the nucleus will divide first. So nuclear division, or the genetic material called nucleoid, or the genetic material divides first to form two equal copies. Like whatever genetic material is there in the parent cell, that will equally divide into both the daughter cells. So first nucleus division occurs, then protoplast, that is protoplasm, divides into two equal parts. That is called division of cytoplasm. First there is division of genetic material, that is nucleus, that is called karyokinesis. After that protoplasm, that is cytoplasm, also divides. So I will try to draw a simple diagram to explain this amitosis. It is a simple type of cell division. Now look at this diagram. Look here. Suppose this one is a simple cell, this is a nucleus. Okay. So first of all, what happens is, this is the membrane, cell membrane. If it is an animal cell, this is the nucleus. Okay. Or plasma membrane, you can write. This is the diagram to show amitosis. Now what happens, there is elongation of the nucleus. The nucleus or the genetic material, nucleoid, it will elongate. See the changes which occurs in the nucleus. And finally, it will divide into two equal parts and separate. The nuclear material is separated, and even the protoplasm also separates. So two daughter cells are formed with equal amount of genetic material, that is DNA, as well as the equal amount of cytoplasm is distributed in both the daughter cells. So these two are called daughter cells, and this one is known as the parent cell. Understood? So it is a simple type of cell division in which there is no spindle fiber formation, there is no centrosome formation, central formation. It occurs in very few steps and it is completed fast. So it is a method of reproduction in lower organisms like bacteria. Microorganisms reproduced by asexual reproduction through this amitosis process also. There is direct division of cell without undergoing any changes in the number and structure of chromosomes. Okay. Now next one is mitosis. Let's come to the topic, mitosis cell division. Okay. Mitosis means the type of cell division which is found in the body cells or the somatic cells. This type of cell division is found in somatic cells or body cells, and it is completed in two steps. Let me draw it first. Mitosis cell division is completed in two steps. What are they? First, division of nucleus occurs, that is called karyokinesis. Karyokinesis. And second one, that is division of cytoplasm occurs. It is called cytokinesis. So karyokinesis means division of nucleus. The nuclear material divides. Okay? Division of nucleus. And this one cytoplasm means division of cytoplasm. Okay? Okay, we have to start with karyokinesis. This karyokinesis or nuclear division is completed in 4 stages, sub-stages. What are they? Number 1, prophase. We have to study all these 4 stages along with the diagram and the important events which occurs during them. Prophase. Second is metaphase. Third one is anaphase. And the fourth one is telophase. Okay, now look at this chart. Mitosis is a simple cell division found in somatic, that is body cells or vegetative cells. That's why it is called vegetative cell division or somatic cell division. And it is completed in 2 steps. The first one is karyokinesis, that is first the nucleus of any cell will divide, and after nuclear division is completed in 4 stages, sub-stages. Name of these 4 sub-stages are prophase, metaphase, anaphase and telophase. After telophase, the division of cytoplasm occurs, which is called cytokinesis. Okay, so one by one now we'll study what then. First, mitosis. It was discovered by, mitosis was described by Strasburger, name of the scientist, in 1870. In 1870, Strasburger described, or first of all, he was to say something about mitosis. Described about mitosis in plant cells. Okay, I'll run it because you have tried. Only mitosis will remain there, the heading. Okay, what is the first point? In 1870, Strasburger, who is Strasburger? Name of the scientist, a German biologist. Strasburger is a German biologist. He described about mitosis where in the plant cells. Now, a few years later, that is in the year 1879, it was in 1870, now the next was in 1879, another German biologist, in 1879, another German biologist only from same country, Fleming. Take the spelling F-L-E-M-M-I-N-G, Fleming. He was the one who described about mitosis in animal cells. Okay. He described it in animal cells. See, he had done it in plant cell, and this person, that is Fleming, had described about the mitosis in animal cell. That is the only difference. But both of them are German biologists only. Then next, who gave this term mitosis? Coined the term. The term mitosis was coined by, means given by Fleming only. He was the one who gave this term mitosis. Coined. It is asked an objective question. Who coined the term? Answer is Fleming coined the term mitosis. In which year? In 1882. Okay. Then next, it is a kind of cell division in all the eukaryotes. You know what is eukaryotes and prokaryotes? Eukaryotes means, eu means true, karyote means nucleus. So all the modern cells are eukaryotic cells because they have got a well-defined nucleus with a nuclear membrane, whereas the primitive cells like bacteria, cyanobacteria, they do not have a well-defined nucleus with a nuclear membrane, and the nuclear material is dispersed in the whole cytoplasm. Even some cell or the membrane-bound cell organelles are also missing in those prokaryotes. Okay, so difference between prokaryotes and eukaryotes, minimum two differences you can learn. Now the next point is, it is found in vegetative cell or somatic cell. This type of cell division occurs or found in somatic and or what, or you can write vegetative cells, hence also known as somatic cell division, hence also known as somatic cell division or vegetative cell division or vegetative cell division. Okay, so what is the last point? Since this type of cell division mitosis is found in the body cells, somatic means body cells, like the cells of our arms, the cells of our legs, isn't it? Cells of our eyes. So somatic, vegetative means those parts of the plant which grow into a new plant, like root, stems, leaves of the plant. They are vegetative parts. So in the cells of vegetative parts or somatic body cells, this type of division will take place, and that's why hence the name is called somatic cell division or also known as vegetative cell division. Who gave this term? The term was given by Fleming in 1882. Who described about it? Strasburger and Fleming in plant cells and animal cells respectively. And both of them were German biologists. Now next, it is a type of reproduction also. This mitosis, it is used by unicellular organisms as a means of reproduction for growth as well as for development also. So it is meant for asexual reproduction, growth and development of living organisms. So last point you can write, it is meant for asexual reproduction. It is meant for asexual reproduction, growth and development in living organisms. Okay, growth and development in living organisms. Growth and development in living organisms. So this method or this type of cell division is used by the living organisms for their asexual reproduction, for their growth also, as well as for their development. Okay. Now let's talk about the different phases of mitosis. You know that mitosis cell division is completed in broadly two main stages or two main parts. What are they? First, there is division of nucleus, which is known as karyokinesis, and it is followed by the division of cytoplasm, which is called cytokinesis. So what are the two main parts or the phases of mitosis? Mitosis cell division is completed in two stages. Number A, karyokinesis, and number B, cytokinesis. Already we have done in the table karyokinesis and cytokinesis. Karyokinesis means division of nucleus, or the nuclear material divides equally into two daughter cells, and that is followed by the division of cytoplasm, which is known as cytokinesis. So one by one we will study about karyokinesis first. Okay. So what are the different parts? Karyokinesis means the division of nucleus. The division of nucleus is called karyokinesis. It is completed in four stages. Number one, prophase. Pro means first, phase means stage. So this is the first stage of nuclear division, prophase. Now what are the changes that occurs in the nucleus of a cell during prophase? That I will draw and explain it here. Okay, prophase is the first stage of nuclear division, during which some remarkable events or changes occurs inside the nucleus. So along with the diagram you can understand. Suppose this is a plant cell or animal cell you take. This is the cell membrane, because animal cell outermost boundary is known as plasma membrane or cell membrane. Inside that cell membrane, or this is the cytoplasm. Then we have the nuclear membrane. Nuclear membrane is there, inside which the nucleus is present. This is the nucleus, this is the membrane. Membrane has pores. Alright? Now inside the nucleus, the genetic material or the chromatin network is present, like thread-like structure. And it also contains nucleolus, since it is an animal cell. So of course, Centrosome is also present. Like this I have drawn centrosome with the centroids. With the aster rays, these are called the aster rays, which are radiating from it. Okay. So this one is known as centrosome. It is the characteristics of animal cell only, not found in plant cell. This one is the nuclear membrane. See, I am trying to draw the diagram clearly so that you can learn and practice for your exam also. This one is called the nucleus. This one is known as nucleolus, the spherical body which is present inside the nucleus. This is called chromatin network. I have drawn a general cell here, chromatin network, which condenses to form the chromosomes. Okay, now during prophase, what are the changes occurs? Number one, this nucleolus and nuclear membrane, both of them, they start disappearing. Means they will vanish, start disappearing during prophase. Means now it is going. Slowly it will disappear because it is not required. So membrane is gone, even the nucleolus will start going, vanishing. Okay, they will also vanish. The second event which occurs during prophase is there is division of centrosome. Centrosome divides to form two centroids. What is the next event? Centrosome divides to form two centroids. Like if this is the centrosome, this is the centrosome, it will divide to form two centroids. Two centroids like this, one more will be formed here. These two are the centroids. This is centrosome. And these two are known as centroids. What are they called? Centroids. These centroids will start moving towards opposite poles. Like one is here, the other one will start moving towards this pole, opposite pole. If it is in the north pole, this one will come to the south pole after division. Means what happens? This centrosome divides, duplicates, it replicates, you can say, isn't it? It makes two copies. One remains there only and one goes to the opposite pole. Means if it is in the north pole, the other one goes to the south pole. And the next event which occurs, this chromatin network, this thread-like network of thread-like genetic material, it condenses, means comes closer to form chromosomes. Condenses to form chromosomes. So first stage, that is, we are talking about the prophase stage. What are the important events which occurs? Condensation of chromosomes and attachment of spindle fibers occur mainly during the prophase. Condensation of chromosomes, that is chromatin network, all these thread-like structures are there. They will come together. Like if you take many threads and put them together, of course the size will become thick, no? Like I have taken one thread, two thread, 3, 4, 5, 6, now see. Many threads are there, now they become thick, ribbon-like. That is called chromosomes. This is what happens during the prophase, that all this chromatin network, which is a thread-like structure, they will come closer and form thick ribbon-like structures which are called chromosomes. Understood now? Now the next stage, it is the longest phase of mitosis. So it's already filled now. Prophase is the longest phase of mitosis. Some points about prophase already I have discussed here, but now I will try to write it down. Prophase is longest phase, means it takes a lot of time to complete prophase stage of cell division. Longest phase of mitosis cell division. Okay, the first point. The second point is the long thin chromatin network or materials condenses. Long thin chromatin network condenses to form short, thick chromosomes. Okay? Earlier the chromatin were long, they were thin. Now they will condense, means come closer, and they will form short and thick chromosomes. Okay? Now the next stage, each chromosome consists of, or it has two sister chromatids. First I will draw and show it also. Each chromosome has two sister chromatids. Now look at the diagram, then you will understand. This is centromere. Okay. Now this centromere means the place where the two sister chromatids are attached. Now this one is one. This one is another one. Okay. Let's draw it nicely. Yeah. So these two are called sister chromatids. See, one chromatid, this is another chromatid, two sister chromatids are present. This together it is called one chromosome. What is this figure of? It is one chromosome. This is the figure of one chromosome. Okay. Now what is this chromosome made up of? It is made up of two sister chromatids. This is one, this is another one. So both of them are called sister chromatids. Okay. So what are these two called? They are called the sister chromatids. Like this lengthwise also you can take one sister chromatid. This is another sister chromatid. Okay. So you can find that there are two chromatids in one chromosome, and both of them are attached at a point. This middle point is called centromere, the place where both the sister chromatids attach with each other. It is called centromere. The place where both the chromatids attach with each other is called centromere. One more structure is here, not mentioned here, kinetochore. What is kinetochore? Kinetochore is the name of the protein part which helps to attach the chromatids with the centromere. It may be asked in the examination also. The next one is, it ends, how the prophase ends. We are talking about prophase. All the other characters of the first stage, prophase. It ends with the disappearance of nuclear membrane and nucleolus. So directly I can write nuclear membrane and nucleolus disappears. Nuclear membrane, already I have shown you in the diagram, and nucleolus disappears. All these are the events of prophase cell division. Then one more point, centrosome. We didn't say right here, but we have already said about it, that centrosome is an important cell organelle found only in animal cells. It is located near the nucleus and it helps in the formation of spindle fibers which are needed for cell division. So this event is the daughter centroids. They start moving towards the opposite poles. Because one centrosome divides to form two centroids, so daughter centroids move to the opposite poles. Daughter centroid move to opposite poles. Any more event is left here? No. And spindle fibers are attached. Like this is the centromere. Here only the spindle fiber is attached. These spindle fibers are formed from the centroid. Suppose if this is the centroid I am drawing here. Okay. So from these radiating rays are produced. No, they are called asters. What are they called? They are known as asters. and they lead to the formation of spindle fibers. So they are known as spindle fibers. S-P-I-N-D-L-E, spindle fibers. The function of these spindle fibers, they will help to pull the chromosomes towards the opposite poles. They attach the chromosomes with the centrioles. Okay? So in the first stage of mitosis cell division, that is karyokinesis, prophase was the first substage. This is the longest phase of mitosis. During this phase of mitosis, the long thin chromatin network which is present inside the nucleus, it will condense, means come together and become short thick chromosomes, means the chromatin network changes into chromosomes by condensation. The next one is each chromosome consists of two chromatids. Both the chromatids are called sister chromatids. What is non-sister chromatid? If I take another chromosome, then they will become non-sister chromatids. I will explain later on about sister and non-sister. The next one is the place where both the chromatids attach, like this one chromatid, this one, where they attach with each other, they attach with this point. This point is called centromere. Centromere is the point where both the chromatids attach with each other. The next one is nuclear membrane and nucleolus disappears. Both of them are present inside the, like nuclear membrane is the outermost cover, it will disappear. Why? Because that now there is extension of the genetic material. Then daughter centrioles move to the opposite poles. And how they are formed? These daughter centrioles are formed by the division of centrosome. Centrosome is the important cell organelle found only in animal cells. It is not present in plant cells. And it helps in the formation of spindle fibers. Okay? Now next we have to study is the metaphase stage. Metaphase is the second stage of karyokinesis, during which all the chromosomes will align themselves, means they will come in the middle. Metaphase, that is in the equatorial region. You know what is equator in geography? You have studied now. This is the second stage. If this is the cell membrane, now there is nuclear material, that is nuclear membrane has already disappeared, so only nucleus is there. Membrane is lost, already gone. I said nucleolus also disappeared, and the centrioles are there towards the opposite poles. Okay, so all the chromosomes, I'll try to make it bigger so that you can understand. You can see clearly. See, this one is centromere. Okay, another chromosome. Only two I am showing here so that it will be easy for you to understand. Okay, now here I will draw the centroid like this. Here also towards the opposite pole, another centroid. Now see, in metaphase stage, these two chromosomes, like this, many pairs of chromosomes may be present, 4, 5, 6 also, more than that. So they will arrange themselves on the equatorial plane. This is the equatorial middle of this. Suppose if this nucleus is like a sphere. So what is the equatorial part? This middle part is called equator. So chromosomes arrange themselves on the equatorial plate. And these spindle fibers which are formed, they attach with the centromere like this. Okay? From here also spindle fibers are formed, and these are attached with the, like this, they are attached with the centromere. Okay, so simple diagram I have drawn. Now let's write what is that. chromosomes align at the equator. That is the main part of metaphase stage. So if you want to learn it in one sentence, chromosomes align. Align means they will come in a single line at the equator. Equator, you know, that is the region. Okay, now what are the events which occurs during metaphase? Number one, it is marked by complete disappearance of nuclear membrane and nucleolus. Now you can see there is no membrane. I have not drawn any membrane here. It's missing, gone, already disappeared. And even the nucleolus which was there inside the nucleus disappeared, gone. So it is marked by, first point you can write, marked by the complete, in prophase stage, remember that nuclear membrane and nucleolus started disappearing. Started means beginning of the event was during prophase only, but they had not fully completed. But now in metaphase stage, it is marked by the complete disappearance of nucleolus and nuclear membrane. Okay, so first important characteristics of metaphase stage, it is marked by complete disappearance of nucleolus and nuclear membrane. And the second one is each chromosome, already we had done this one, that chromosomes arrange themselves at the equator, forming the metaphase plate. This is called metaphase plate. Chromosomes arrange themselves at the equator. Chromosomes arrange themselves at the equator, forming equatorial plate. Forming equatorial plate. Okay. Now, any more event which happens? Yeah, we can say that the centroids have already reached towards the poles, and they help in the formation of spindle fibers which are attached to the centromere. So third characteristics of metaphase stage, centroids reach at the opposite poles. Already I said centroids reach at the opposite poles. Now any more? The last one, already I said chromosomes, spindle fibers attached at the centromere region. Here already explained. Spindle fibers attached at the centromere. Spindle fibers attached at the centromere region. Okay, in the middle, this part. Spindle fibers attached with them. So this is spindle fibers. This one is chromosome. This one is centroid. If you have to label all the parts, this is known as nucleus. This one is known as asters. These arrows which are shown on this one, just like sunlight, sun rays are there, these are called asters or astral rays. Aster or astral rays also we write, R-A-Y-S rays. So in this way we have completed the second or also metaphase stage. So metaphase is the second stage of karyokinesis cell division, in which the chromosomes align at the equator. You know what is equator? Now what are some of the events which occurs? It is marked by complete disappearance of nucleolus and nuclear membrane. Nuclear membrane which was present outside the nucleus, it has disappeared, and the nucleolus, the dense spherical body which was present inside this nucleus, has also disappeared. Okay, completely disappeared. Number two, chromosomes arrange themselves at the equator. These chromosomes try to remain on the equatorial plate, and they form equatorial plate. So this region is called equatorial plate. Equatorial plate means the part of the equator in which all the chromosomes arrange themselves in a line. Then next one is centroids reach at the opposite pole. These are the centroids. They move towards opposite poles. Both of them. Like if this is in the north pole, this will be in the south pole. That is called opposite poles. And the spindle fibers attached. These are the spindle fibers which are formed by the asters. From the centroids only they are formed by the elongation of these astral rays. So these spindle fibers attach the centroids with the centromeres. Okay, so these are the events which occur. Now next we have to study about anaphase and telophase. Okay, so next stage we have to do was the third one, that is anaphase. Now, what is the important characteristics of anaphase stage? What happens during this? I will write it. Chromatids separate in the bracket here only. Chromatid will separate. What is chromatid? Chromatids are formed from the chromosomes only. Now, one by one, along with the diagram, we'll try to learn what are the important events which occurs during anaphase. First, let's draw the diagram only. What is this? This one is nucleus. Okay. Now, I'll draw the centrosomes. In simple, I'm just trying to draw. Here also one centroid. Okay. Now, let's see what is the position of centroids. They are in the opposite poles. Now, you'll find that the centromere is here. 1, 2, 3. 3 chromosomes I am trying to draw. 1, 2, 3. Let's not make it very complicated. What are they? They are the centromeres. And these are the chromatids. See how the arms I am drawing. Arms are pointed towards the equatorial point, this one. Whereas the centromere is facing this one. See, I am drawing dot, dot, dot. Okay, here also. What are they? They are the spindle fibers. And this one, the arms which I am showing, they are the chromatids. So like this, try to draw it nicely. Here also in the same way. Okay, now look at this diagram. This diagram, what is it? You will find that earlier there was this centromere, no? I will just draw it here. Centromere is there, in which one chromatid. This is one chromatid. This one another chromatid. Both the chromatids are present. That's why they are called sister chromatids. Okay. Now during this stage, anaphase stage, there is division of centromere. The centromere will split. Now from here, if I will cut them, split. Then see, 1, 1, 2 are formed. Okay. Now these two chromatids separated. Can you see from here? Due to splitting of centromere, the centromere was split, and then two chromatids were formed. These chromatids are also called as sometimes as chromosomes also. So they are attached with the spindle fibers. These are the spindle fibers. And when the spindle fibers contract, like from the centroids, due to contraction of spindle fibers, they are pushed towards the opposite poles. Like this, these chromatids will move towards the north pole, and these chromatids in the southern hemisphere, they will move towards the south pole, towards this centroid. Means contraction of spindle fiber pull the chromatid away towards opposite poles. I hope you have understood this one. So let's write what are the important characters. It is marked by splitting of centromere into two functional centromeres. This anaphase is marked by splitting of centromere into two functional centromeres. Both of them are working, that's why functional centromeres. Then second, spindle fibers contract and pull the chromatids towards the opposite poles. That is an important part of anaphase stage. So you can write it. Chromatids separate from each other. Now see, both the chromatids were present together earlier. Now they separate from each other and they are being dragged towards the opposite poles. So chromatids separate from each other and move towards opposite poles. Chromatids separate from each other and move towards opposite poles. Why they are moving towards the opposite poles? Due to contraction of spindle fibers. Okay, this is the forces which is acting to push them away from each other. Anymore, chromosomes at this stage appear of different shape. Like, see, what is the shape of this one? Like, if I put the centromere here, what is the shape of this chromatid? It appears like L shape. Okay. What about this one? If centromere is here, it appears like V shape. Okay, now next. Centromere is here, one chromatid here, another one is like this. It appears like J shape. So chromosomes are of different shapes depending on the position of centromere. So chromosomes appear as L, V, J and I shape. Chromosomes appears as L, V, J or I shaped. Chromosomes appear as L, V, J or I shapes, and they during the movement or towards the poles. This process is very fast. So one more characteristic we can write: rapid process. Anaphase is a fast process. Okay. Any more? Finished? Now last stage we have to do, that is telophase stage. In the telophase stage, what happens is all those events which occur during prophase. I'll just try to draw it here only so that it is completed. Whatever happened in the prophase stage, it will be reverse of that. Like nuclear membrane disappeared in telophase, the nuclear membrane starts reappearing. So although it is the diagram of anaphase, now I am trying to draw telophase here only. So nuclear membrane reappears. I am not writing, I will just say nuclear membrane reappears. What it is? This is nuclear membrane. Nuclear membrane reappears. Even the nucleolus also reappears. Some slight changes we have to do now here, because it is the last stage now, telophase stage. Okay. So nucleolus reappears, the dense spherical body, and now whatever chromosomes were there, now they will uncoil to form the chromatin network is formed. See, earlier chromosomes are there. Now these chromosomes will open, unwind the threads which were wounded. Now they will open to form individual threads. That is, chromatin network is formed, and then there is deepening of membrane. The nuclear membrane will deepen further. They will help in the separation of this one, the depression. Like in case of animal cell, a furrow is formed in the cell membrane. In the nuclear membrane, the furrow will be formed that will divide the nuclear content into two equal parts, and both the parts will contain equal amount of genetic material. So what happens in telophase? The chromosomes relax. What are the separated chromosomes? They will arrive at the poles. So all the chromosomes will be towards the poles. Okay? It is making us confused. Let's do like this. I will draw some separate diagram. Okay? Now look here. Now this is one pole. Here all the chromosomes are present. see another pole. Here also all the chromosomes have reached. Okay, they have reached towards the opposite poles. This one. Earlier in anaphase they were moving, but here in the telophase stage, what happens? The chromosomes reach the opposite poles. So one point you can write: chromosomes reach opposite poles. They have reached opposite poles already in telophase stage. And second event, a new nuclear membrane reforms. Already I had said nuclear membrane is reformed. In that previous diagram I had drawn, it was not clear. Okay, so nuclear membrane reappears. Nuclear membrane and nucleolus reappears. Then chromosomes uncoil to form chromatin network. Once again the chromatin network, same like prophase stage, is established. Okay, and centrosome, like whatever centroids were there, now once again the cleavage appears. Chromosomes uncoil to form the chromatin, already I said. Now cleavage, this one we will leave it. Cleavage means what? That happens during the, like now the nuclear division is almost complete. This is the last stage of nuclear division. So after that cytoplasmic division has to begin. In case of plant cell, a cell plate is formed in the middle. Okay, in animal cell, a cleavage or furrow is formed. Cleavage, C-L-E-A-V-A-G. Cleavage means is formed. The deepening occurs. Like if it is the animal cell, this cleavage will be there, like this. Here also, this cleavage will form, which will further deepen, and then that will cut it into two equal halves. Similarly, in the plant cell, the cell plate is formed, which will divide it into two equal halves. So in the next class, we can study about the cytokinesis and continue with the further topics. Till then, thank you.